We have a very exciting panel now with Peter Jones, Scott Harper, and Yi-Wen Chen. They will be talking about different therapeutic strategies that researchers are using to try to tackle FSHD. So they will introduce what these ideas are, and then we'll definitely open it to questions from you.
All right, so the theme of this session is therapeutic approaches, and so this is the simplified version of our scientific meeting that we've put together. The focus you've heard about today is DUX4 can exist as an mRNA and a protein, and most therapeutic approaches are going to, one way or another, impact this pathway. And so I can introduce Dr. Scott Harper here from Ohio State and Nationwide Children's Hospital, and his work is actually focusing on this area, doing some technology to knock down the DUX4 full-length mRNA and potentially even the going after the protein through translational inhibition. And also Dr. Yi-Wen Chen from, now officially your hospital is?
Children's Hospital, D.C.
Okay, Children's Hospital, D.C. has a different type of technology to do the same, to attack the mRNA through an antisense technology. My lab specifically, we're upstream of this. We're actually trying to shut down the expression of DUX4 by going after the regulation of the pathogenic gene. And then there's all these downstream markers down here, the things that we know that can happen once you express DUX4 and that can be found in FSHD cells and FSHD patients. And the reason I want to put this up here, and we're just going to actually just leave it up here, leave this as the focus, is essentially, for FSHD therapeutics, you know, all the folks on DUX4, there are a lot of avenues for intervention, therapeutic intervention. We don't know what's going to work for individual patients. Some patients may respond differently to different approaches. The other thing is there's other approaches that we don't work on, that other researchers here work on, and there's a number of companies that have their own therapeutic approaches. And so I guess what I would kind of leave with or lead into with is there are a lot of potential avenues for therapy. There are a lot of companies with their own particular approaches to therapies. It wasn't like this 5 years ago. We have a target, a really good target, DUX4. This allows us to look upstream as a second set of targets, downstream as a third set of targets. And so this is actually really good news. Okay, and we only need one to work. It would be nice if multiple approaches worked. You notice there's a lot of differences in patients around. Maybe different patients will respond differently. You've heard a little bit about different repeat sizes having different effects. Maybe different repeat sizes will look different. So the good news is a lot of avenues for therapy, a lot of approaches, and actually a lot of companies interested now in getting involved in having funded FSHD research programs. And so that's pretty much our presentation. We're going to open it up for questions, and any questions you all may have about therapeutic approaches, we'd be happy to take them.
I've got two questions. First, what will happen, or do we know what's going to happen if we turn off DUX4? Will the muscle repair? Will we gain strength? Will it just stop progression? What's testing has been done in that area?
I'm not sure we know. In my opinion, I think there's a chance that there could be some restoration, but I think at a minimum we can arrest progression, and I think that those are questions that we still don't really have the answers to. I don't know what my other panelists think.
So I think the knocking down DUX4 is the first to stop the muscle get further damaged. So whether it can recover afterwards really depends on what's the state of the muscle at that point. So if we consider the muscle still have its normal regeneration ability, and that seems the case for most of the patients. So when you think about if your exercise can actually help you to get stronger, that means your muscle stem cells still there, and they are still doing the job. They are maintaining the muscle. They can even make the muscle stronger, bigger. Hypertrophy, in this case, like one of the trials. So it's likely they will recover, but like Scott said, it really depends on what happens. So in case like if the muscle is severely damaged or it's completely replaced by fibrosis or fatty tissue, and in that case, whether it can recover really need to be studied further to be able to know. So I think both other researchers here, they actually have animal models, and those actually will be good.
So the good news is it's a slowly progressing disease, so slowing it down is actually a pretty good step. Stopping progression, especially this gets back to getting genetically tested in family members, knowing when therapies comes, the less damage when you start the therapies, the better. Restoration is a more difficult thing, but I don't think that's off the table. So it's, but I personally, I'm confident that we will be able to slow down or stop progression.
My question for the three of you guys is, so these are three very different, points, I guess, in DUX4, D4, Z4. So why do you believe that your research is the reason, or that would cure or treat FSH, and why you believe you're doing what you're doing? And the second part of the question is, if we were to come out with therapeutic targets, would a benefit of all three together, or would it be just one portion of it, or would it be two of the three? Like, what do you think?
So speaking for myself, I think that the potential, the strategies that Peter laid out at the beginning all have promise. And for me, so what my lab is doing, we are trying to reduce expression of the messenger RNA, the DUX4 messenger RNA, and try to prevent that from either being expressed or made into DUX4 protein, which we think is the toxic element. And the reason that my lab decided to start doing that is simply because we had expertise in it. There were some practical approaches there. And so we knew how to accomplish that. And we knew how to deliver our essentially what we're doing is gene therapy. So we knew how to deliver our gene therapy to muscles in an efficient way. And so that's why we took that approach. There was very much a practical aspect to it, but I don't think that means that the other approaches that others are trying to develop would be any more or less effective. And to your second question, I also think that the notion of using combinatorial therapies at different points in the expression pathway for DUX4 would probably be useful, and I think that's something we should be considering.
Well, I think that gets back to the previous question, which is stopping it is maybe pretty good, and then can you actually recover? So there's benefits to that. And that actually second Scott's question. It's our expertise. We got into FSHD research because my graduate student was a patient and said, this is an epigenetic disease. I happen to be expertise in epigenetics. And so we started, we're using our expertise. I think you'll find a lot of people using, and as more people get into the field, thanks actually in large part to the FSH Society, you're getting more expertise, which is actually opening up more avenues for therapy as we're learning more about it as the more expertise comes in. And so I have nothing against the downstream approaches whatsoever. Our expertise is looking upstream. It's an epigenetic disease. If we can return the epigenetic status back to normal, that's a great therapeutic strategy, in my opinion. But doesn't say anything about other approaches. And combinatorial, we just need one to work. And I'm not smart enough to tell you which one is going to work right now. So.
Yeah, I definitely agree with both of them. I think it's first depends on what we are good at, what we understand more. So we choose the ones that we think we can contribute the best. And then the other thing is, everybody can only do that much research. So it's actually a team effort. Everybody's doing their own part, and hopefully, and also that will increase the chance that something actually pan out at the end.
Yeah, you don't want to focus in too early. Right now we want to be broad and a lot of approaches and figure out what's going to go, so absolutely.
If I could just make a follow-up comment on that before the next question. I came to my first FSH Society research meeting 10 years ago as a new assistant professor starting my lab up, and I came just as an observer. And at that time, just 10 years ago, there was still a whole lot we didn't know about mechanism, about how the disease was caused. And now we are fast forward 10 years later, and we have a whole session now on therapy development. It's remarkable to see the progress. Not only that, there was a handful of people at that meeting I went to 10 years ago, and this was, I think, probably the most well-attended scientific meeting that I've been at in the last 10 years. So I think there's much hope and progress being made.
Testing. So first off, thanks for being here, guys. But my question is about supplements. Is there any one supplement or combination that would be helpful at, CoQ10, creatine, glutamine, etc.?
I can comment on what's actually ongoing now, like for the clinical trials that's available. So I know there is a creating trials for early onset, FSHD ongoing, maybe recruiting at this moment in Australia, and there has been studies on antioxidant, vitamin C, vitamin E, zinc, and selenium as a supplement ongoing in France. I don't think there's any trial in terms of supplements that's ongoing here, but definitely these studies are conducted because there are some evidence show these may be beneficial. For example, for FSHD, a defect in oxidative stress responses that's being reported before. So that's actually one of the reasons a trial is ongoing. But I don't think there's any conclusion at this moment. So there's no scientific conclusion that's a specific combination or any specific ones that's better. But I think very soon we will have some reports.
So the million-dollar question is, how long do you think this is going to take to get there?
We'll let Scott answer that question.
Give me the hard questions. I'm always reluctant to make a timetable. I think there is reason for hope, as I mentioned. I think we're making a lot of progress, but science isn't linear, and we often, most of the things we do unfortunately fail, and we have to step back and figure out, regroup, figure out what we did wrong or what we could tweak. And so I think, I guess to be cautious, I'm a little reluctant to put a timetable on anything. But I can tell you, just as an example, just a few years ago, I guess 5 or 6 years ago, well, I guess I would say maybe 2007 was I think when DUX4 really started to come on the scene, and then it gained momentum as being a gene target. And in that intervening time, we now have a number of mouse models and cell models and other zebrafish and frog models of the disease that express DUX4. And that sounds like a long time, but 2007 to now, but in scientific terms, that's a remarkable amount of progress. And I bring that up simply because these types of models and systems are absolutely required to generate therapies. So I think the point I would just want to stress is that it's hard for me to say personally when we're going to have something online, but at the same time, the amount of progress we've made in a short time is very hope-inducing.
And to me, the really big thing that shows not linear, I think we went to the same first meeting. There's just no industry interest. We didn't have a target. The field was just, we didn't have such direction. Now, it's astounding how much pharmaceutical company interest there is now. And part of this, I'll get back to testing. A larger patient population, more interest from pharmaceutical companies get tested. Getting organized for clinical trials. Because the reason the next talks are going to be about getting organized for clinical trials is we're not just blowing smoke. Clinical trials are coming. Okay, there's a lot of interest. There's a lot more researchers in academic labs. There's a lot more industry research. It's not linear. We can't give you a date. I agree with Scott. But boy, if you can see it from our perspective, it is really, things are really picking up steam. And so I'm actually very encouraged, or I'd be working on something else.
And the other point is that the field has grown. You know, there was a narrow body of researchers doing a certain amount of work, and as we're understanding more about the field, we're bringing more minds to it. So I think that helps as well.
So all of us have heard over the last couple of years about the wonderful CRISPR-Cas system that everybody thinks has the ability to make magic transformations in people's genomes. So Peter, you've published on it, and it's been widely discussed in the Facebook group, and I'm sure all of our patients attending want to know, is CRISPR-Cas therapy for our somatic cells a pathway that you think is realistic?
A pathway that's realistic. Everybody, I assume, knows what he's talking about with CRISPR-Cas. Everyone said people have heard of this term. It's what's going to...
Head shaking.
No, really. So people are living on Mars. No. Made the front page of the New York Times on December 31st. Not a great time. So this is genome editing. Okay, this is genome editing or genome modulation. Basically, there's now a mechanism where we can specifically target regions of pretty much any genome, including the human genome. And you can change, in the lab, we can change the gene sequence. We can also target regulatory molecules. And what our lab published is that we could actually, in the lab, in primary FSHD muscle cells, turn off the DUX4 gene using a virally delivered CRISPR-Cas system. My instructor in my lab, Karis Hamida, will be have a presentation of this at the gala today. The big question is, can this actually become a therapy? We cure lots of stuff in the lab. We cure lots of mice, a lot of diseases, but we need to cure people. The good news that one of the reasons we're pursuing CRISPR-Cas is because the entire world is pursuing CRISPR-Cas, not for FSHD, but for other things. And this gets to an earlier question. What do advances for other diseases have to do with FSHD? Technological advances in other diseases, other muscle diseases, almost anything can really advance FSHD research. And I'm convinced that there's so much potential for CRISPR-Cas technology. It's very inefficient right now. It's difficult to deliver, but the world is working on this, and for a lot of diseases. And I think there is potential. And when the smart people figure it out how to deliver this therapeutically, the FSHD field is going to be ready. Okay, so I actually have faith in it because, again, there's a lot you can work on. So we're working on things that we truly believe have therapeutic potential. When's it going to get to the clinic? I don't know. Probably further off than a lot of other things. But, you know, there's the worldwide effort to get CRISPR-Cas technology to the clinic. So I'm a believer in it, if that answers your question.
Perhaps this is related to the question you just answered or tried to answer as well as you could. But would any of you or perhaps other scientists in the audience who might still be here address whether there is any work being done with stem cell transplants and FSHD or other dystrophies where the results might be applicable? And I think one of your colleagues, you seem to be getting ready to answer, your earlier comments seem to suggest that perhaps some of your work was related to that area.
Is Michael Kaiba in the audience by chance?
No.
So, well, I'll answer as best I can. I don't think any of us are stem cell experts. There've been the short answer to your question is yes. There is an ongoing body of research, not necessarily focused on FSHD, but in muscle therapy and muscle biology in general, to develop ways to restore muscle using stem cells. And I don't think the short version of the story is that I don't think they're quite ready yet, and that you can get widespread engraftment at this point. But I know there's a huge effort to do it.
Exactly.
Hi, I have a question. The question is, has anybody got any information as to what the triggering effect is within your body from being non-symptomatic for 52 years of your life, and then all of a sudden you're 52 and you can't do anything like you used to? Is there any idea what's the triggering effect to becoming symptomatic?
So that is a spectacular question.
Thank you.
And because I look at that as essentially there's an avenue for therapy. right? If we know there's a reason that, my student was playing soccer when he was 15 and 16, and then 18 he's getting weaker, and then he progresses much faster than his brother, who actually is very slowly. And are there triggers? What are the individual differences? Are there lifestyle changes? And I think that's something that the natural history studies should come out of that, hopefully, to see what, as you follow disease progression through people. There are a lot of anecdotal things about hormones, for example, or age, going through stressful events in life, but to actually have a scientific documentation of these things. And if we could identify triggers, I think, if there, it would be the easiest thing. If there were lifestyle changes or some other avenues of intervention, that would be very beneficial to know. But I don't know of any, actually.
I just have one more quick question. I'm wondering, just from a scientific point of view, could it be the over-exercise or under? Like, I'm a court reporter in criminal court, okay, and I can't lift my arms anymore, but I have full use of my hands and fingers, so we go 300 words a minute. And I've been doing this for 40 years. So I'm wondering if the lack of use of a certain limb or muscle group versus not using it perhaps would have some kind of a clue. I don't know. Just wondering.
Is someone a physician?
So is it about over
Larry, are you? Larry?
I think for muscular dystrophy, at least at this moment, there is no evidence that. Is here somebody here?
We have some over there.
Robbie,
We have a couple of physicians over in the corner that might be able to answer that.
Maybe we can save that for the next session, actually. They're coming up after us. These we're basic researchers. I think that's really for the MDs.
I'm Amanda here in the back. I just thought I'd mention that the company that I work for, Genea, does have an active drug development pipeline for FSHD, but we also have a muscle cell transplantation pipeline because we can take pluripotent stem cells and differentiate them to muscle cells. So that's one of the sort of arms of our FSHD therapeutics development program that we have, and could obviously be applicable to other diseases as well. So feel free to come and talk to me about it if you're interested.
We should have known that. Sorry, Amanda.
I think we might have time for one last question.
Hello everyone, my name is Isham. I'm from Paris, France, so thank you for your indulgence for my poor English. I would just wanted to confirm what said Dr. Chen about supplementation. Basically, I'm part of this clinical trial in France in which they give us some vitamin C, E. I mean, I could talk to you more about this if anyone is interested. But it brings the question about, last slide, about oxidative stress. And my question is, did you study this oxidative stress on the mice?
For the mouse model. So at this point, we are testing, for example, our antisense oligos. So when we collect the muscles, we do plan to check the oxidative stress response genes. That's being shown that's affected in patients, but we haven't done that